Hi, my name is Fred Davies, and I'm a professor in the Department of Horticultural Sciences at Texas A&M University in College Station, Texas. I'm going to be talking today about principles of propagation by cuttings, and I'm going to go through some of the anatomical and physiological aspects of propagating cuttings and rooting and adventitious bud and shoot formation. And I'm also going to give a second presentation as well too on phytohormones and how these plant growth regulators are used in cutting propagation systems as well too. I might mention that I am a Florida Gator. I got my PhD from the Department of Environmental Horticulture here at the University of Florida. And one of the things that brought me to Florida as a student was I was very much interested in plant growth and development, and still am. And the whole area of plant growth and development is pretty unique when it comes to propagation systems. And in propagation, what you're doing is you're taking morphology, you're taking botanical aspects, you're taking physiology, you're taking molecular biology, you're doing all these things to go ahead and manipulate plant materials to produce very, very unique plants which can have a very high cash value. And cutting propagation is one of the most important forms that we are able to go ahead and regenerate plant materials. Cutting propagation is the clonal multiplication of plants with propagules of stems, leaves, or roots. You have already learned what a clone is, and basically a clone is a genetic copy. And we use clones all the time in horticulture. I can't think of an area in agriculture where clones are used more extensively than in horticulture. Some advantages of using these clonal systems is we can produce plants which have superior characteristics. It could be a fruit crop, for instance, which has certain desirable fruit characteristics we're looking for. It could also be in ornamentals, the form or the color or the shape of a plant. And because we can clonally regenerate plants, because we can make these genetic copies, which are all uniform, that allows us to go ahead and command a much higher value, a much higher price for the product we're producing. There's more cost involved in doing it, but the product produced is far superior. And again, that's one of the areas that makes horticulture very unique. Cutting propagation is the most important means of clonal regeneration of many horticultural crops, ornamentals, fruits, nuts, vegetables. And a lot of times, for instance, in vegetable crops, we normally think of them as just being seed produced. But if you look at some of the really important vegetable crops around the world, for instance, potatoes and what have you, and sweet potatoes, and also yucca, and a number of other different types of species, they are asexually clonally regenerated. On the upper right-hand corner there, you see a picture of a poinsettia, which is being rooted in that cube. That cube was split in half so you could see the root system. But poinsettia is an example of a multi-million-dollar industry around the world. And the reason we're able to go ahead and have that industry is because we can asexually root these cuttings and go ahead and produce the certain cultivars that we are trying to go ahead and market. Another area also of cutting propagation has to be with adventitious buds and shoots. And adventitious buds and shoots are going to be very important from the standpoint of regenerating new plants from leaf cuttings or also from root cuttings. And when you get right down to it, an adventitious bud and shoot, they arise from any part plant other than terminal, lateral, or latent buds on stems. They also form irregularly on older portions of a plant and not on the stem tip or in the leaf axils. And what you'll notice in the upper left-hand corner is you have a bud, which is what we call an adventitious bud, and that adventitious bud will ultimately go ahead and develop into a shoot. And what we refer to that adventitious bud as being an embryonic shoot. Essentially, it has all the characteristics of that shoot. It is a shoot. And as that adventitious bud goes ahead and develops, it goes ahead and forms that shoot system. And it's with that shoot system that you're able to go ahead and get multiple shoots to occur. So in the case of begonias, which you happen to be looking at in these pictures, that's very important from the standpoint of regenerating new plants through cutting propagation. Adventitious roots, of course, are really important in cuttings. And with adventitious roots, we're working with stem cuttings, or what you're looking at is a picture of what we call a leaf bud cutting. And what you've got is a small leaf, you've got an axillary bud, and you've got a small piece of the shoot system as well there too. Adventitious roots arise from any stem part other than by normal development and ontogeny of the seedling root and its branches. Adventitious roots can also be regenerated from root prune seedlings. And if you think about it, in the ornamentals nursery here in the state of Florida or in Texas where I work, if you look at the different methods which are used in ornamental propagation for producing plants, probably about 80 to 90%, depending on the crop species, are going to be regenerated through the use of cuttings, particularly stem cuttings. So stem cuttings is a very, very important area then for being able to regenerate plant materials. Let's go ahead and take a look at stem anatomy and how stem anatomy becomes important from the standpoint of rooting. These are Ficus pumila, and Ficus pumila is also known as creeping fig. It's grown here in the great state of Florida, and what you have is what we call a mature form and a juvenile form. And the mature form is characteristic of it being able to go ahead and form fruit. The juvenile form is more vine-like. It tends to go ahead and climb. It also tends to be much easier to root as well too. Looking at the stem anatomy, if we go ahead and this is a cross-section on the left-hand corner of the mature material, and by cross-section basically what we're doing is we're taking a piece of that stem and we're just cutting across with a microtome, and then what we've done is we've gone ahead and we've stained different cells and tissue there. And if we take a look at the juvenile material, there are some differences that occur here. Looking at some of the structures that we find in the cross-section of the stem, we have what's known as the periderm, and the periderm is the outside layer. When a seedling begins, for instance, it has what's known as an epidermis. And if it happens to be a woody perennial plant, that epidermis will later go ahead and develop into a periderm. So both the juvenile and the mature form have periderms on the outside. They also have what are known as sclerids, and sclerids are basically lignified structures, and they help give the plant support. If you think about it, what a plant's doing all the time is it's fighting gravity. And if it didn't have certain cells which gave it certain rigidity, those plants would just collapse under the shear force of gravity. So as a plant becomes more mature, it's going to go ahead and produce more sclerids, as you can see on the left-hand corner there. And again, the reason for that is structural support. Why am I spending time talking about sclerids? Well, when we start to talk about rooting, some of the myths about rooting is that when a plant becomes more mature, these sclerids actually form barriers which prevent the rooting process. And in most plant systems, that's simply not the case. Sclerids are not a limiting factor to that rooting process. We also have what's known as the cortex. And with the mature to the left, you can see the area of the cortex there. And if we go ahead and we take a look at the juvenile, you can see the area of the cortex there as well too. Those large circular cells are parenchyma cells. And I'm going to be talking more about parenchyma cells later on, but all cells initially start out as parenchyma cells, and then they may become very highly specialized and develop for certain cell types that they go ahead and form. The phloem is another important area as well too, and this is the area of the phloem both with the mature and the juvenile material. Those blue structures that you're seeing there are what we call phloem fibers. And again, those phloem fibers help to contribute to the rigidity, the stiffness of that stem to enable it to be able to stay upright and create the certain form that it does. The area of the phloem becomes very important because, as you know, the phloem gets involved in the translocation of carbohydrates. The phloem is also important too because auxins are also going to be translocated in the phloem as well too. So the phloem becomes a very important site for where roots will originate from. In terms of adventitious roots, there are different types of adventitious roots. We have what are known as preformed roots. A good example of preformed roots, for instance, if you look at the slide to the right, preformed roots very typically will form in the axil of the plant itself. So on the shoot structure itself, sometimes there are roots which are formed in the normal ontogeny, the normal development of that shoot system. Salix, which is also known as willow, which is a woody plant species, also has primordium, which are formed. And again, we refer to these as being preformed. Some of the ivy, for instance, pathos to your left there, again in the area of the leaf ax, in the area of the where you've got an axillary bud occurring there, that's where you're also going to find roots that are preformed in the normal development of that shoot system. We also have what are known as wound-induced roots, and these are formed de novo. And all de novo means just formed anew. And here's an example of de novo rooting at the base of that leaf bud cutting. You notice the roots are coming out at the base. And what's happened is when we took that leaf bud cutting, we essentially did some things to start to stimulate the rooting process. And those roots are going to be formed de novo. It has nothing to do with the normal ontogeny, the normal development of that shoot system. It's roots that form after we have gone ahead and taken those cuttings. And probably de novo rooting, with the exception of something like pathos or some easier rooting stuff, is the most common way that we will have roots forming from cuttings. In terms of de novo adventitious root formation, there are 4 different stages involved in that process. The first stage is known as dedifferentiation or remeristemation of parenchyma cells. And the whole idea behind this is we are challenging parenchyma cells which have differentiated to go ahead and differentiate or dedifferentiate into another type of cell groups. And what we are doing in de novo adventitious root formation is we are forming a new meristematic area where a meristem did not previously exist. And a meristem, again, is an area where there's very active cell division going on, and that cell division becomes very specialized in the formation of an adventitious root or the formation of an adventitious bud. In both those situations, we are creating new meristems. But here we're talking about rooting. So the first step is known as dedifferentiation or remeristemation. The second stage is what's known as initiation of slightly organized cell groups. We refer to those as being root initials. And what's happening is the meristematic area is becoming more and more organized. We start out with one cell initially, there's more cell division that takes place, and there's more cell division that goes on, and we start to see the early beginnings of a meristematic area. Ultimately, we'll have a full-blown root primordia that differentiates in the cutting itself, and that again will be occurring in the phloem and also developing into the cortical area, the cortex as well too. And the last stage is known as elongation or extension of root primordia. One of the things you're going to learn in propagation is that some plants are easier to root than others. And the reason we have some plants being easier to root than others is the fact that they are able to go through these first 2 stages, this dedifferentiation and the initiation of slightly organized cell groups. They're able to go ahead and dedifferentiate and form root potential. They have what we call the genetic potential. And all plants in the beginning have that genetic potential, but there may be certain physiological changes, and sometimes we refer to those as being epigenetic changes. There may be certain changes that go on, and a plant species, a woody plant species, as it becomes physiologically mature, may lose that ability to dedifferentiate and go ahead and form root initials. And that's a really important area because in forestry, for instance, there's a big interest in being able to clonally regenerate plants. If you think of an elite specimen ornamental plant, by the time we know it's really got unique values, it's going to be physiologically mature, and that means our success of taking a cutting is going to be very low. So again, the key point here is there are 4 stages involved in de novo adventitious root formation, but the first 2 stages, dedifferentiation and initiation, are the most important. If a cutting can go ahead and get through those first 2 stages, chances are it's going to go ahead and form a root successfully. Looking at dedifferentiation or remeristemation of parenchyma cells, what we have here is a cross-section of a stem. And one of the things we see in this dedifferentiation process, again, this is with a woody perennial plant here, is we have increased vascular cambial activity taking place. And where this arrow points, that's the area of the vascular cambium. And the vascular cambium gets involved in producing both the xylem and the phloem. And the xylem is that tissue at the bottom. You can see larger type cells. They're kind of reddish and what have you. and then above the vascular cambium you're getting into the phloem area. So there's very typically increased vascular cambial activity that takes place in the early events of rooting. What we have is a cross-section to the right, and we're going to be looking at what's known as an anticlinal division. And that anticlinal division is going to be taking place with phloem ray parenchyma, and that's where this whole process of de novo rooting is taking place. It's actually starting in this parenchyma cell, which has gone ahead and it started to divide. And we call it an anticlinal division because it's perpendicular to the circumference of that shoot that we've gone ahead and we made a section in. So again, if we look at that parenchyma cell, again it's in the phloem area, and below it at the very bottom we're in the actual xylem itself, and just below where the anticlinal division is taking place you see some other cells which are starting to divide. Well, that's all part of the phloem and also the cambial area. So this becomes an area where there's a lot of activity going on. Phloem ray parenchyma cells, why are they so important? As I mentioned earlier, all cells are initially parenchyma cells. And what may happen, certain parenchyma cells may become very specialized. They may go ahead and form sclerids. Remember those sclerids were those really deep red cells? Also fibers. And sclerids and fibers, when they become functional, lack a nucleus. And they're very important for what they do, but because they lack a nucleus, they cannot undergo further cell division. There's what we call totipotency, and totipotency was talked about earlier in the course. And totipotency basically means it's the genetic potential that plant species have of being able to regenerate a whole new plant clonally by actually taking just a single cell. That's called totipotency. Well, totipotency is also important because in forming adventitious roots, we have to have phloem parenchyma cells or some form of parenchyma cells which has the genetic potential, the genetic capability of being able to go through all these cellular events to form a meristematic area. As I mentioned, parenchyma cells can develop into other cell types, and again what we're trying to do in the formation of roots is we are trying to challenge these parenchyma cells to revert to meristematic cells in de novo rooting. And that's not so easy to do all the time. The phloem, another important area for the phloem and why parenchyma cells in the phloem play a big role in rooting is that we sometimes refer to them as a loading zone, kind of like a feeding area when you get right down to it. And the whole idea behind a loading zone is it's an area that's really rich in carbohydrates because this whole process of photosynthesis is going on. These metabolites are coming down and they're being translocated in the phloem. The phloem has special cells and stuff like that which actually get involved in helping to enhance these metabolites moving around and what have you. So it's a very nutrient-rich area. Also, auxins are important, and auxins get involved in stimulating rooting. Well, auxins are translocated through phloem parenchyma cells. So phloem parenchyma cells are a really logical area to have roots occurring. You've got cells which potentially have the capability of undergoing cell division to start this meristematic process. You also have an area that's really rich in carbohydrates and metabolites because, again, there's going to be very high respiration levels that go on. This is a really energy demanding type of process to go on to form these meristems. And then you also have auxins which are being translocated, and they become important again in stimulating the whole rooting process. The second process that goes on is the initiation of slightly organized cell groups, and we refer to those as being root initials. And here's a classic example of a root initial. This is in the phloem area. And this root initial, again, we're seeing more and more cells starting to divide. So it started out from one cell, and there's continual cell division that's going on, and slowly this meristematic area is becoming more organized. It's not yet a full-blown root primordium, but it's becoming more organized. And again, this kind of outlines where this whole root initial is so you can get an idea of what it really looks like. And it's pretty cool to see these things. In terms of root primordium development, this is the third stage that goes on, and here we have a full-blown primordium that occurs in the stem section. There's going to be the cortex, and what you can also see here is the root cap, and the root cap are these cells which are staining very dark here, And you all know what a root cap looks like, for instance, in the corn seedling, where if you look under a microscope, it has a very distinct structure to it. Well, you can actually see root caps which are developing internally in the stem as this primordium is going ahead and becoming more developed. There are a bunch of meristematic cells as well that occur here. Closer look at these meristematic cells. One of the things you'll notice on the right-hand side there, where that arrow is pointing to, is that they're very dark, they're very dense. The nucleus is very large in proportion to the rest of the cell. Also, where that arrow is pointing to, you'll notice that there are 4 sides to that cell, or what we're seeing in our just two-dimensional view of that. Those 4 sides are equal. We call those isodiametric. And what that means is that this is just a classic example of what meristematic cells look like. And again, we've really challenged and forced this cutting to go ahead and go through this process. The last stage is the elongation or extension of roots, with roots coming out through the actual cutting itself. And here we have what's known as a longitudinal section. And what that means is we kind of hold the cutting upright, and we cut through it longitudinally. And what you'll notice is there's a root primordium, and that primordium is well developed. It's also starting to push out through the sclerids. Those sclerids are those red cells that I showed earlier as well too. And again, if you look at how that's pushing out, those sclerids are really not a barrier. They're really not preventing elongation occurring. It's only in a few plant species we see that process going on where there can actually be a barrier that occurs. If you look at the root primordium, particularly towards the tip of that, some of the work that's been done by Margaret McCulley and others, for instance, have shown that actually the root tip, which is forming again, pushing out to the cortical area, has a lot of enzymes. Those enzymes get involved in lyasing. They actually will go ahead and dissolve some of the cortical cells. And there's a tremendous amount of hydraulic pressure that also goes on as that primordium pushes out, and it goes right through those sclerids. And here's another view of that as well too, where you can see the sclerids. There's 2 primordium there. The arrow just is pointing